guest. Sorry. Um, and she is Dr. Mariana Gómez-Schiavon. She is an associate researcher at the International Laboratory for Human Genome Research and has complete interesting work at the junction of biology and mathematics. For example, with the feedback control of biological molecules with proportional integral derivative, a concept often used in engineering, such that a biological system can return to a set point after a perturbation.
Thank you. Thank you very much. First of all, I just want to say that it is an honor to be here. I'm so glad that you are trying to change the culture, to really make science a more like a part of daily lives of young people, particularly girls. But I think everybody needs science in their lives, and math is beautiful. But all types of science are good to have. So thank you for letting me contribute a little bit to your effort. Do you want me to start telling you a little bit of my trajectory, or how do you want to do this? I don't have a specific plan, so.
If you'd like, we can ask you a few questions.
Okay,
first.
perfect.
Okay. Well, first we'll start with something general. What is it like to be a woman studying biology, mathematics, etc., and other scientific domains?
I mean, it's weird, right? Because we have always been a woman, like all of us. And then you grow up as a girl, and then you become a woman, and there are like just so many things that we normalize are part of our daily lives, and we just think that is the way it is. So it's almost impossible, for good and for bad, it's almost impossible to imagine what it feels to be someone else. How many of the things that we experience and live are because of me, because of just chance, because I am a woman, right? So I always struggle with that definition. I don't know. A part of me, I mean, there are like so many aspects that I don't know even where to start, but a part of me is just really proud, you know, like proud of being a woman in science, proud. Like last time that I was talking with some of you in the mathematics Olympiad roundtable, I was saying like it's important to own your privilege, to own what you know, like it might have made your life better and other people might not have, as education, for instance. That is definitely a privilege that I have. But on the other hand, also I think we need to own our struggles. And so at the same time that I'm very proud of just being a woman in science, I recognize there are like certain barriers that you have to overcome time and over again. And I think Karen was saying just a moment ago, like how you have to prove all the time that you are good enough. We get used to it, but I honestly believe it's not the same if you're not a woman. And it might be harder, again, on your privilege, it might be harder for other groups, particularly with diverse gender identities. It might be even harder for people like them. But in general, it's just like we have to prove ourselves all the time. And for me, being a biology, but a quantitative biologist, like trying to do math, I think especially when I was starting, like people will assume that I didn't know math. And I have to prove over and over again that no, I do. I probably know more than you because you are a biologist, and this is something I really like. I'm also a biologist, right? But I have been doing this all my career, like from where I started in high school until now, I do math. So I know. I'm not a mathematician, but I know, right? But with time, I feel, I don't know if it's because I'm more confident or just because it comes with authority. But as I have been, you know, like climbing the ladder and just getting in these higher and higher positions in the academic career, I don't feel that anymore. I feel like people actually respect what I have to say in math. So that would be the optimistic side of it. I think it gets better. There are like other things, of course, that keep happening, but it gets better. Yeah, sorry.
Thank you so much for your answer. I'm glad to hear that. So our next question is, do you believe that scientific research is most impactful when it unites multiple scientific domains? Like for example, your research paper on the analysis of proportional integral derivative control of a proportional integral derivative controller with biological molecules? If so, could you please tell us a bit more about what makes this particular paper novel? For example, what are the coolest ways in which you believe PID control could be used to regulate the functions of biological molecules?
Yeah, thank you so much. That's such an specialized question. I would be happy to answer. So first, the part of joining fields. I think science in general just benefits from diversity. Like, and we sometimes think about like, and now I'm getting into the social again, but I'm gonna go back to the science, I promise. But sometimes we think about diversity just as saying like, well, we all need an opportunity. We need to be fair and just, and all those things are of course very important, but just being pragmatic, science benefit from different perspectives. And you never know where those perspectives might come from, right? So when we standardize that scientist just means that you have to be fill the blank, you know, like it's like you have to be a certain type or certain gender or certain nationality. That's bad for science because we don't know where that perspective might come from. And saying that, and going back to the science, the same happens with the fields. Like we can accomplish a lot of things just specializing in one little thing. Like I'm gonna do microbiology, and microbiology is not little, but just to put an example, right? Like you're just gonna focus on that and keep asking the same type of questions until you get it. That's good because science also needs persistence and, you know, like you just keep trying. That's also important, but that will give us like a very, very narrow sense of what is out there. So we need to ask different questions. So when you mix fields, that's what happens. I'm going to ask a question with a different perspective because I'm thinking about it from a mathematical side. I'm going to ask a different question because I'm thinking from a sociological side, right? Like all those little things, especially if we get to work together, which sometimes is hard because surprisingly, or not that much maybe, different scientists, we basically speak different languages. Like if I'm talking with like a pure physicist, like we don't understand each other. We say different, like completely different languages, right? But then there are people like me who are like in the middle. And that's also why it's important that I'm not a mathematician. I wasn't trained as a mathematician. I was always trained in the middle, but that's probably one of my biggest strengths in what I do, because I do speak biology. And then I do speak a little bit of math. And then I can do something different and I can also communicate. So I can pull together the biology side and the math side. And hopefully that's what I aspire to at least, ask a different type of question. So yes, I do think they benefit. I wouldn't say that everybody should be doing mix. That's okay. People have different interests and diversity is what we need, right? Now about in that paper. So the novelty there is just to say that this PID control is like a fundamental tool or concept that you have in engineering. Like it just basically has the basic components that you need to have good control. And I think it's important to specify what control means in this context. And it basically means repressing disturbances. Like anything, you want to do certain specific behavior, and then you have things going around you, or maybe even inside the system, right? It could be outside or it could be inside the system, but things that you cannot, that you don't want to happen, but they happen. And they are kind of disturbing your behavior, and you want to pull it back to where is the desired behavior. Like you can think about a plane, flying, you want to fly at the same altitude, you don't want to be like this, right? Like that would not be nice for the passengers and also probably not safe. So you want to stay as much as possible in the same altitude. So you have a control system to measure, like if there is some change in the pressure and you get down a little bit, you do something to go up again and things like that. Or you can think about the thermostat in a house. Like if you want to keep your house, or maybe like in Mexico, the house thing is not a good example because none of us have a thermostat in our houses. That's not a thing. But maybe in the fridge, right? You want to keep your fridge at 4 Celsius because that's the good temperature to keep all your cheese safe, right? So if you want, like if you open the door and the cold goes outside and it's maybe 40 Celsius outside because you are like in a very hot summer day in, I don't know, in the beach. And then you open and it gets warm inside, then something senses, and that's an important part of control, something senses that it got a little bit warmer inside the fridge and you turn it on. And maybe that is the winter and it's getting really cold and the fridge needs to know that, oh, now it's really cold outside, so I don't have to invest that much energy to keep it cold. And then like you do it less, right? So that is control. You are noticing that something is changing outside and you put it back together. Now, there are many ways you can do that. In general, like I say, you need the component of sensing and then sending the corrective signal. In the case of the thermostat, it would be like if it is getting warmer, you're sensing the temperature goes up, then you turn on the ventilator to pull it down. And if the temperature goes down, then maybe you stop the ventilator so it can go up again, right? Like you're playing with those two things. So sensing and correcting, that is a standard. But other than that, how you sense and how you correct can be different things. And particularly the proportional integral derivative control, what it's doing is either sending a corrective signal that is proportional to the error, that is actually taking the integral of the error, or that is taking the derivative of the error. And they have different properties in how the system responds. And that is very well studied in engineering and control theory. That is actually more mathematical than anything else. But the thing is that all those hardcore theory is using mostly some assumptions as your system is linear, or it can behave almost linear in this context. And one important thing about this linearity is that it doesn't saturate, like you have always the same response and you can work with the functions that way. And biology is never like that. Biology is complicated precisely because almost everything saturates. Almost everything is non-linear by definition. So you never have like this response, you have more like curved things. It's a little harder to predict when they curve, how they respond. You might think like it works in certain range of values, but then you move, it completely breaks. At least in the way we think about it and in the way we do synthetic biology, because that paper is also in the field of synthetic biology. That is when we take molecular components, parts of the cells, and we use them to build something that we want, right? Like a little machine inside a cell, but we are building. But what is surprising is that actually biology does control very well. We just don't understand how. So part of the idea with that paper is it's just a little step, right? Is that we are very far away from solving this problem of how actually biology does it so well, and in which ways it differs from what we know from control theory and engineering. But what this paper does is, if you wanted to do something like this, and you wanted to do it with molecules that saturate and respond non-linearly, how would you do it? And we propose a specific design where even if it is just a model, it's only mathematics, we are not building it for that paper. Is still like saying like, well, you will need a promoter here. And I'm like starting to use language from biology that you might not know, but promoter is the part that regulates the expression of a gene. So you might need like a promoter of this form here. You might need that this gene actually produces a molecule that is called a transcription factor that goes and recognizes this other promoter, right? You start like putting the different parts and the links of how they regulate each other to actually accomplish what formally will be defined as a proportional derivative and integral controller of the error signal. And the signal in there is just expression level of something. You want to keep the expression of a gene around a specific value, and you want it to be able to sense when it gets away from that value. And I'm sorry if I got too technical. I can answer more or try to explain it better.
We love that. Thank you so much. It's so interesting how you applied a concept that is like typically or like classically used in engineering and robotics to biology. So thank you for informing us about that. I see that somebody left a question in the chat for you. What do you like most about your career?
For me, it's funny because I never ever consider studying mathematics, but for me, what I like more is just the mathematics of it. I love the patterns. I love how like you can have like just one, two components. Components usually means genes in our case, but if they are connected in the right way, they just have these amazing emergent properties, these non-intuitive behaviors that are so beautiful, and they're just like little pieces of what it is actually the complexity of biology, right? So I guess for me, I always was fascinated by how biology is so complicated and beautiful, and at the same time so robust. Like every single time, like a mother gets pregnant, every single time it makes a little human, right? It doesn't make a little weird thing. It just makes like it does the same thing. Like it works. Biology works, right? And for me, like is the patterns of it that we can explain with mathematics what makes me passionate. But I guess that is what I like most about my science. But I guess my career, it involves many other things, my job, my daily job, many other things. And for that, I will have to say, it gets connected in a way, is actually explaining these things to students or to people. In general, I don't care if they are young, old, experienced, non-experienced, but when I get to say these like beautiful things, and you can see the face of the other person just lighting it up, like when they actually see it, it is just the most beautiful thing for me. So I love teaching. I love interacting with my students. I have a few, right now is 3 students, undergrad students that are working with me, and I just talking with them about their projects, seeing how there are like, you know, their ideas are growing and things that I haven't thought before. They are just thinking and sharing with me. Like all that part is for me the most enjoyable part of my daily work.
Thank you so much. Another question we have for you is, which one of your research papers are you, or projects are you most proud of, and why?
Okay, so it's always hard to choose between your babies, right? So I will have to say that my PhD paper, the one in plus computational biology, it is about the selection or the evolution of epigenetic switches. It is so full of jargon, and I apologize for that. Like there is just not better way to explain this, and that's why I guess it shouldn't be my favorite, because it's so heavy. But it is also the work that inspired what I'm doing in my lab. Whenever you start your own lab, you have to propose what is going to be your research line, and my research line is fundamentally inspired by this paper. And also this paper has so many things in it that I'm just like proud of it. But I can tell you a little bit more about the paper. So the idea is that we have, so over genetics, right? So we all have our genetic information that is in our DNA. But for instance, just as an example, because it happens at all levels of life, from bacteria to us, all the cells in your body has exactly, or in principle, has exactly the same DNA. It has the same genetic information. And still, you see that the cells that forms your eyes that are capable of sensing light are completely different from the cells that maybe are in your skin, right? Like not only morphologically, also in function, also like it doesn't matter how you measure them, they are completely different, and they still have the same genetic information. So what does make the difference? Well, one, regulation. The regulation is different between the genes involved, but the way they keep that regulation, and when you reproduce a cell, when the cell in your skin reproduces, it doesn't produce like magically an eye, right? You don't get an eye coming out of your hand. You have more skin. So that memory of being able to pass that regulation information is what we call epigenetics. Now, there is a question of how epigenetics came to be, like in the first place. And then when you think about bacteria that are not doing that, they don't have like different organs and different functions. Well, they have different functions, that's kind of the point, but they don't have like these structures. And still we see that they do have this memory and they change. And more importantly, they change stochastically. That means kind of randomly, but not completely random, but it has a pattern. I'm saying so many technical things. I'm so sorry about that. I don't know how to explain it better. But anyway, so bacteria do have also this epigenetic memory. And we observe that they do it to basically try to deal with fluctuating environments. The environment for all of us is all the time changing, and we have to be able to keep up in order to survive. But for a little bacteria, it might be, a thing of life or death. Like if you are growing really happy and then it comes an antibiotic and it kills you. So you have to be prepared for some things that you cannot really know when it's going to happen. And then this epigenetic thing can help you, this stochastic or random epigenetic transitions can help you to be prepared for these situations. But it costs energy, like they cannot grow as happy, because if all of them were prepared all the time, they would be sacrificing all the time because, oh, the antibiotic might come, right? But if they never prepare, then the antibiotic comes and they kill all of them. So you have to balance this thing of, I'm going to prepare part of my population and I'm going to let other part of my population grow happy. This phenomena is called bet hedging, because in English, like you are like just balancing your bets of like, oh, I'm betting is not coming versus I'm betting is coming, and you are playing with that. And it results that everything is connected, right? To be able to do this bet hedging mechanism, you have an epigenetic switch, but we want to understand how this epigenetic switch, when, in which circumstances specifically, is favored and why. And if it can appear from nowhere, right? From zero. That's how it connects with other epigenetic memory. We want to see if it connects from zero. Now, this might sound like a very specific, tiny thing, like, oh yeah, it's cool, but why do you care? Well, it's because we not only care about this example, it's because this case is exemplifying how evolution can happen in an emergent property, in something that, if you think like the epigenetic switching is not one gene. You cannot have like one gene and that is epigenetic switching. Epigenetic switching is a behavior, a dynamic behavior that, for instance, we can only explain mathematically, right? So how are you selecting through evolution a mathematical property? That is something that, to start, is kind of very abstract. And in that paper, we kind of put it, like something that you can touch. And you say, well, if you want to have this mathematical property that in biology called epigenetic switching, in mathematics, we call that B-stability. And B-stability requires positive feedback. So we choose the simplest gene regulatory system that can that can give us positive feedback, and we let it evolve. And we let it evolve in a circumstance where we are gonna be introducing an stressor, an antibiotic, something is changing. It doesn't really matter, but we are introducing it. But the individuals that we are modeling, they are not real, right? Everything is in the computer, but like the individuals don't know when it's coming. I know because I control it, but they don't have a way to know it. And we observe that, yeah, this mathematical property, just as itself, it is actually selected. So what it tells you about like the connection between the molecular side of it, our gene regulatory circuit with positive feedback, and the evolution and how they connect to these mathematical properties, to me, is just beautiful. And it might sound abstract to you. Believe me, it sounds abstract also to biologists. Like this paper was a little hard to publish because of that, because it's kind of a different idea. And that's also why I'm so proud of it. But that's still what we are trying to do in the lab, just with different little things. But it's still this idea of, we want to understand evolution, or how natural selection acts, or where these properties of the we think a framework that allows us to really, define these questions. And we think when we contribute to that, it's going to change with problems. That is a little ambitious of my side, but I think it's fair. It's fair to dream, right? Like I think we can make a contribution.
you. And lastly, we could ask you, what advice would you give minorities who are pursuing STEM career?
Wow, yeah, that's hard. So the basic is just don't give up. But at the same time, I say that I feel unfair because it can be really hard, and it's really easy to say from the privilege, right? Like, don't give up. But I think more than an advice for them, I think I would like to make a call for the rest of us. Like, we have a responsibility to be there for the other, because it's fair, because it makes us better, like all of us as a community, and because you never know when you're going to be on the other side, right? Like, what does it mean to be a minority? Here in Mexico, I'm privileged. That's the reality. I had education, I had family, I had, like, I cannot complain about anything. But in the US, I was an immigrant, and I was a Mexican immigrant. And maybe I didn't suffer as many as other Mexican immigrants because there is racism, and I don't have the phenotype that is as rejected as others, but still, I suffer a little bit of that, right? So you never know when you're going to be in the other side. And I think it's just also part of that, but more than anything, because it makes us better. But so my advice to minorities is just keep dreaming and keep fighting as much as you can, and just maybe try to find allies, people who want to help you. Try to communicate as much as possible what you're going through. And again, like, when I say those advices, is a little unfair because that is extra work for them. Like, oh, you also have to educate people around you. Yeah, okay, not do it alone, but at least the people you trust, because sometimes we get blind for privilege and we don't realize what the other person is going through. So at least you people you trust, communicate what you're going through, and humanize scientists. We need to humanize scientists as much as possible so we all feel welcome. Yeah, sorry, I don't have a better, I struggle with this. It is an important topic.
Thank you for your eloquent response.
Good. Okay.
Thank you so much for your time and sharing your amazing experiences and advice and your research. Thank you so much for your time.
No, thank you for inviting me. This was really fun.
Thanks. And thank you so much for all the audience. This can't be do without you. And see you in the next series. Thank you so much.
Bye-bye.